Hello, my name is Autumn Burlide, and today we're going to discuss my proposed expansion for ASU's Mother database. So you might be asking, what is Mother? Mother is an open access resource started by ASU with goals to further understanding of reproductive health and mechanisms involved in a variety of species. In my professional life, I work at Tufts Veterinary School, and I have developed a liking to studying cancer biology and veterinary models that have potential implications for human medicine as well. So that is my basis for studying this needs-based proposal. I think this is a big reach for a project, but this data could have strong implications. The goal of this proposal is to aid veterinary diagnoses and the understanding of the molecular drivers behind ovarian cancers in macaques. The other benefit of this project is that since it would be under Mother, the information would be open access, not behind any sort of paywall. Anyone in the world could access this data and potentially develop drugs or therapies that target proteins which are commonly implicated in ovarian cancers. Therefore, my null hypothesis is that all protein expression is equal amongst cancerous and non-cancerous cells alike, and that my alternative hypothesis is that there actually is a difference between these groups. The methods for this determination will be briefly discussed later. So we're currently limited in a few ways to perform the entirety of my proposed expansion. For example, the H&E slides that are on the Zielinski laboratory and the IHC slides are actually from non-cancerous macaque ovaries. So these slides could actually be used for morphological controls, but they couldn't be used for cancerous slides. And they also didn't collect any protein at the time of removal. I'm not sure if the removal was during a necropsy or during just a sterilization of the macaques. But regardless, protein wasn't maintained or retrieved at that time. And there are also no current resources for protein assay data on the Mother database. So we would therefore aim to acquire fresh ovaries from at least 3 female macaques that are known to have malignant neoplasms of the ovaries and 3 healthy ovaries so that stats can be performed. So say we get these ovaries. This is what we would need to do. First, we would collect the fresh tissue from the macaques, and that will be prepared according to whatever operating protocol the submitting laboratory is. But in general, it should follow these guidelines. At the time of removal, either during necropsy or sterilization, the ovary should be used for microtomy, should be fixed in 4% PFA, which is paraformaldehyde, for the appropriate amount of time. Usually that can be anywhere from like a few hours to a few days. And then once it's fixed, then you can store it in PBS if you're not going to immediately cryosection it. Additionally, if funding should allow, unfortunately, spatial transcriptomics is incredibly expensive, but it's also incredibly insightful. So, should funding allow, several slides with representative sections of tumors should be used for spatial transcriptomics. The other ovary should be immediately homogenized and processed for protein analysis. For the fun part, which is our wet bench work, we would use the Pierce assay and Western blotting techniques. In figure 4, you'll actually see an example of a Pierce assay plate, which will have a darker color when the concentration is higher and lighter when it's lower. So usually the first 2 rows, 1 and 2, are controls, which are typically regarded as standards, with either increasing or decreasing concentration, so that you'll see an actually really cool gradient as you go down or up. So in the first 2 rows in figure 4, you can see that it gradually goes from a really dark purple color to a lighter green color. And that should represent that those first 2 are probably the really dense ones or the really concentrated ones. And then the lower ones are probably either just PBS or some very light concentration. I actually really like to perform these assays. I think it's really fun to get a visual when you do science. That's why I also really love immunofluorescence. Anyways, then we can also do a protein expression analysis by doing Western blotting. These can be used to measure densitometry, and they'll be uploaded as either a TIFF or a JPEG. So what you'll do is once you're done performing your Western blot, you'll image your Western blot, and that's what you'll see here in figure 5. And then you can take that image and upload it to a software like ImageJ, and you get the densitometry values. And then from there, you can add that to a CSV, and both of these results will be in the form of CSVs. So these can be added to Mother in the form of zip files containing the CSVs. And images from the Western blot densitometry can also be included for transparency, because a lot of publications do require the full image of the Western blot. And last but not least, I did want to briefly review some of my sources. So source 1 was where I pulled the information regarding commonly affected proteins in ovarian cancer, and Geomics is the brand of spatial transcriptomics that I would suggest we use. They used to be owned by NanoString, but then they got bought out by Brooker. Um, but I've had good experience with that company before. Additionally, we just plug the Mother database there and some other resources for the Pierce assay and Western blotting, some other scientific resources, and the Zelinsky lab, which is where I was looking for the H&E slides and the IHC slides of the ovarian cancers. All in all, thank you so much for watching my project. I think it would be really cool if funding would allow, or if resources were available for this. Thank you so much.